Hello and welcome to Beyond Biotech, the podcast from Labiotech. And this is episode number 4. I'm Jim Cornell, and it's been an unusual week in that it was July the 4th on Monday, which means absolutely no news coming out of the US industry, but that seems to now extend to globally these days because no one wants to put out any news because it won't get coverage in the US. So it was a slow start to the week, and that meant Tuesday was really busy. And on Wednesday, my holiday started and my wife's family arrived, and I couldn't find any parking at the airport, which was a great start. While I may be away from work, of course the podcast needs to come out, so here it is. I'm not even really sure I know how to take a complete break. And I don't really mind. I do have to say it's a lot quicker when there aren't any technical glitches, and today also sees the podcast debut for Labiotech's Liza Laws, who did one of the interviews this week. There's been a bit of a heat wave in Europe, and that even crept its way north to Scotland, where we had a day of 27 yesterday, which is just creeping into the 80s. I know that that's not really high in many countries, but it turns heads here. Actually, it burns a lot of heads. And I speak as someone who is follically challenged. But yes, I do also wear a hat. So let's take a quick look at who our guests are on this episode of Beyond Biotech. We talk to Helen Zha, Assistant Professor of Chemical and Biological Engineering and a member of the Center for Biotechnology and Interdisciplinary Studies at Rensselaer Polytechnic Institute in the US. We also speak with Klaas Zuideveld, CEO of Versameb, and we have one interview with two people, and that is Aamir Ehsan, CEO and Medical Director of CorePath, and also with Celina Villa, Medical Director of the Biopharma Division at CorePath. All right, it's time to run through some of the news you may have missed over at Labiotech.eu, or at least some of it. US biotech financing has dropped by 46% in Q1 compared to 2021, according to a new report. A nanoparticle can kill cancer cells and aid muscle relaxation. And Brie Bio has taken up an option on a hepatitis treatment. We had an article on metagenomics, a look at the biggest biotech investments in June, and AstraZeneca added to its treatment options with a $100 million deal. There's positive news from a trial for a drug to treat a major depressive disorder. Mogrify and Astellas are to address hearing loss, and Novartis Pharmaceutical UK launched a health tech investor partnership for cardiovascular care. EditForce and MTPC are to work together on gene therapy for CNS. Sanofi Global Health launched 30 non-profit medicines around the world, and Destiny Pharma announced the start of a new oral mucositis research program. A new discovery center to house scientists in India has been set up. We look at 5 advanced microbiome players, and Finnish and US companies have formed an exclusive agreement for the development of a sustained-release drug. Biotech food colorant startup Phytolon has sealed $14.5 million in funding. 3 companies have joined forces to tackle antimicrobial resistance, and you can read all of these and a lot more at Labiotech.eu. You can also check out this podcast at Labiotech.eu, and you can send us press releases there as well. And while there, you can read some of our evergreen stories. And that doesn't mean that the leaves don't fall off. It actually means there are articles that are relevant and informative whenever you read them. So that brings us to our first interview. World Continence Week took place at the end of June, and one company giving a presentation on stress urinary incontinence during the event was Versameb. We spoke with the company's CEO, Klaas Zuideveld. So I wonder if you could first tell me about the company and its origins.
Absolutely. Versameb was founded in 2017 by Friedrich Metzger here in Basel, Switzerland, really as an RNA company with the idea to use RNA to produce therapeutic proteins. Friedrich was previously the head of rare disease drug discovery at Roche, worked a lot in sort of muscle physiology, a number of other indications, and really saw the opportunity to, with a different modality to actually create some drugs. So that's how the company was founded. He subsequently built a team. Initially, quite a few scientists, took on a business development person, and actually joined in 2020 really as a chief development officer, and actually took over as the CEO end of 2020.
I wonder what kind of things you're working on addressing in terms of conditions.
I think the company really started around the platforms. We have a platform technology. The platform is really geared to make mRNA more effective. And with that, I think there's two focus areas. One is being able to increase the potency so that with less material or with small amounts, you can actually get the therapeutic levels of the protein. And the second is to make the mRNA more effective. And that's one of the directions we've taken there, is our ability of actually building a technology which allows us to combine mRNA with siRNAs. And similar to increasing the potency for mRNAs, we've been able to increase the potency for siRNAs in this context as well. In terms of conditions, we're applying it to hard to treat diseases. And that's a fairly broad concept, of course. But with that, we've actually chosen two sort of indication areas for now, urology and oncology. And really coming from the perspective that I think mRNA lends itself particularly well for sort of regenerative medicine. It is what we're applying in urology, in stress urinary incontinence. I can say a little bit more about that later. And the other area, oncology. Oncology is, of course, a very popular area also for mRNA. But there's lots of sort of, in addition to a number of proteins you would like to bring to expression, there's lots of proteins you would like to suppress, particularly sort of oncogenes. And I think that's where it sort of lends itself particularly well for the platform where we can actually increase the potency of the siRNAs. So in oncology, we're working glioblastoma, which is obviously very hard to treat area, and then also non-muscle invasive bladder cancer. Non-muscle invasive bladder cancer being one of the areas which lags behind in terms of therapeutic development. So not much has happened in years, and some of the treatments are fairly dated.
Could you tell me a little bit about the Verse Agile platform?
Yeah, absolutely. No, the Versameb platform it's actually a collection of 5 patent families, with the core 2 actually being around the sort of the technologies I mentioned before. We actually have a deep expertise in modifying some of the sequence code to the actual RNA sequence code on the front and the back end, or the flanking regions of the coding region. And by doing that, for example, increase the expression, we can enhance the translocation of the mRNA to the endosplenic reticulum, and also increase the secretion, so that on a per milligram basis, we can actually increase the amount of protein, increasing the potency of the mRNA. This is really built around an algorithm which will optimize the signaling protein. Similarly, enhancing or working on a linker technology, we've been able to actually combine mRNA with siRNA in a single construct. So we're actually putting the siRNAs, typically when you administer siRNAs, they typically get cleaned up by the immune system, but by the body fairly rapidly, by sort of putting them into sort of a Trojan horse, like the mRNA, packaging them in the same construct. We can smuggle them into the cell and actually have, using the cell's sort of natural existing cleavage process, we can cleave them, and then the mRNA can do its function, the siRNA can actually do its function, suppressing whatever gene we want to suppress. So we've been able to do this reliably for up to about 4 proteins. So we can modulate the expression of one protein and then decrease the expression of another one at the same time. Of course, being able to target 4 proteins increases the number of targets and number of constructs we can develop hugely. So that's really sort of makes up the platform. The other 3 patents are around sort of reducing immunogenicity. There's some specific applications in oncology and urology, which you've also been able to protect. That actually makes up the platform itself. That said, of course, we can expand beyond that. And we've been in discussions with a number of companies to actually look at other disease areas such as aging, immunology, or sort of inflammation. So I think there's quite a way to go here.
Could you tell me a little bit about the presentation on the lead candidate, VMB100, for stress urinary incontinence?
So this week is actually World Incontinence Week. I mean, there's actually there's a number of different forms of incontinence. Stress urinary incontinence is one of the larger forms of incontinence. It really involves the involuntary loss of urine due to stress on the pelvic floor or on the lower stomach. It affects both men and women, but it affects women, significantly more than men. So we see about 1 in 3 women suffering from stress urinary incontinence at one point in their lives. The typical treatment sort of goes from, say, conservative treatments like physiotherapy, doing pelvic floor exercise, and then when that doesn't work, you're immediately looking at sort of surgery or, say, bulking agents. And so there's no real drug treatment in this space. There's one treatment, which is really an antidepressant, which women typically don't like to take very much. There's no drug treatment in this space. And that's where we saw the opportunity, saying, okay, there's actually a very large market with no competition, together with actual issue being, of course, that the 2 leading causes of stress urinary incontinence are, say, natural childbirth and aging. And what happens during natural childbirth is that the stress on the pelvic floor actually causes damage to the urinary sphincter. It will repair by itself if women are young, but of course one of the issues, of course, that by the time women have children nowadays, say around the age of, say, 30 in the Western world, women are already well on their way down onto actually the levels of IGF-1 in their body, which is responsible for repairing muscle damage, actually is decreasing. So that what we see is that about half the women which are low IGF-1 people, they will actually not recover by themselves. And of course, as IGF-1 levels decrease further, women don't actually recover from incontinence as they get older either. So that's that protein which we try to bring to expression in the urinary sphincter itself. And we've actually generated some very nice data in animals. And there's actually only one animal model for stress and incontinence in the rat, which simulates a pregnancy. We've been able to show that we can actually regenerate the muscle tissue and also regain its function very nicely. So that's what key opinion leaders are very excited about. So we've actually developed a dark product profile, tested this with a number of key opinion leaders around the world. And that's with that, we're actually going into the clinic next year.
And in terms of the treatment, how does it work? How is it administered?
So mRNA actually doesn't lend itself very well for systemic application. Of course, a lot of the success from the mRNA, also with the vaccines in the last few years, is really all local administration. So we're also going with a local administration. So it applied directly to the urinary sphincter. The urinary sphincter is actually a very small organ. Applying it directly ensures that we get the drug to the site of action and it can actually do its work. The application itself, we're looking to test in a first-in-human study in patients. It would involve one or maybe two injections, and that should be sufficient to actually regenerate and restore the function of the urinary sphincter itself.
Once that's done, that's the end of it? You don't have to redo this every so often?
Well, I mean, in animals, I think we see a very long effects. Of course, the animals don't live as long as humans do. Of course, we anticipate that if women have a second pregnancy or if they age, it might reoccur. That said, we do anticipate to see a very long-lasting clinical effect, which should last at least a year, 2-year period.
And so where are you at when it comes to the whole process?
We've completed our pharmacological studies. We have proof of concept, and we have quite a bit of data. I mean, one of our advisors, also Margot de Meester. Margot de Meester is actually the one who actually developed the animal model for stress and incontinence. So I've been working with her. We've completed our first GLP toxicology study, so that's done. That was the main study, actually looking for toxicology. The second study will start later this year, looking for the actual route of administration, using the injection device, which we'll use to inject material into the urinary sphincter. We do it in a larger animal, administer that. We have our production ongoing. Production of mRNA is still challenging. It's difficult. There's 2 steps to this. We've recently started a collaboration with a company which can do a fully synthetic approach, which is very attractive. So we're getting to very high levels of purity. And of course, purity is important in a therapeutic setting because you don't want to have any immunogenicity or any other issues. So that's ongoing. So by the end of the year, we should have sufficient clinical materials to actually go into the clinic next year. We've had interactions with the FDA. We had our pre-IND meeting. But the main thing there is that it gives us a clear path towards being able to start our first in-human study in patients, in women with stress and incontinence after natural childbirth. At the moment, we're raising financing, as many startups, obviously, really with the objective to finance our clinical program. So we're very excited to move that forward.
Are there any other areas that you're working on right now?
So we're also working on oncology, and there we're actually focusing on glioblastoma and non-muscle invasive bladder cancer. The program with glioblastoma, we're actually collaborating with a university. That's still very much in the sort of pharmacology discovery process. We have about 11 molecules which are actually looking to select the best one to move forward. But there's still a number of animal experiments we're looking at doing and ultimately moving that into sort of explant studies before making a decision which molecule to take into the clinic. Similar to non-muscle invasive bladder cancer, this really came out of the discussions we've been having with urologists on the stress and incontinence side. We see a huge unmet medical need there as well, where one of the best treatments is still the so-called BCG, or the flooding of the bladder with basically tuberculosis bacteria, which has a lot of sort of logistical issues. Plus, I think we believe we can actually do one better, both being able to stimulate the immune system, as well as being able to shut down a number of oncogenes at the same time. That we're advancing ourselves. I have to say, we do have some interest from pharmaceutical companies which we're exploring to partner with them in this area. But again, that's also more in the pharmacology discovery stage.
So are collaborations very important to the company?
Absolutely, yeah. No, I think particularly in the financing environment has of course changed with changing market conditions. I think then having collaborations becomes much more important and attractive for companies.
Is there anything else that you wanted to cover?
It's a competitive space. We believe we're very differentiated by focusing also more on the pharmacodynamic properties of the mRNA and hope to prove that in the clinic very soon.
Next, it's to the podcast interview debut of our own Liza Laws. She spoke with Aamir Ehsan, CEO and Medical Director of CorePath, about the recent collaboration with CISL on cancer diagnostics. And later on in the interview, you will also hear from the company's Medical Director of the Biopharma Division at CorePath, and that is Celina Villa.
This is Dr. Aamir Ehsan. I'm the CEO and Medical Director of CorePath Laboratories. CorePath is a cancer diagnostic lab which is headquartered in San Antonio, Texas, USA. Basically, what CorePath does is we do a full spectrum of diagnostic testing for cancer patients. We are CLIA accredited and CAP accredited, which means that we are accredited by College of American Pathologists, which is the highest standard for any laboratory to maintain in the United States. We are staffed by academically trained board-certified pathologists, PhD doctors, and clinical lab scientists. So every day we are taking care of cancer patients, making the diagnosis for all kinds of cancers. As you know that these days, precision medicine is the really key thing for cancer patients. And for precision medicine, we need to have a precise testing and precise diagnosis. And that's what we do here at CorePath.
And so how do you sort of interact with patients? Do people get referrals to you, or the work you do gets passed to specific hospitals?
So we have a broad national-based networking through our sales and marketing team that reach out to various hospitals and oncology practices and other pathology practices. Patient samples are coming from all parts of the United States through Texas for us to process these patient samples. So basically, cancer samples that have been obtained in different hospitals and different oncology centers are coming through FedEx to here at CorePath to be tested.
If we could move on to the exciting news, is the partnership with CISL, just sort of like the history and the background, and how you came to come together.
One of the CorePath mission has been to take care of research and development and biopharma industry. And it was kind of a national partnership. Benke, who leads the Benke Group, introduced us to CISL Biotechnology, the public traded company from UK. They had a novel protein that they have identified as CISL Biotechnology, and they were interested in rolling out this protein testing for early detection of lung cancer globally. And one of the largest markets is in the United States. So through our networking, they reach out to us looking for partnership in USA in a lab which offers cancer diagnostic testing, which is CLIA and CAP accredited. So we have a couple of communications. We look at the concept of novel protein, and it just happened to be a very natural partnership with CISL Biotechnology for the betterment of making life better for cancer patients.
Will you be sort of looking to target other countries where it's also a huge problem, or for the moment, are you sort of concentrating on where you are?
Our niche and our presence is in the United States. CISL Biotechnology is reaching out to other countries globally. And as you know that every country and each country have their unique challenges of compliance and regulation. So we are focusing more in the North American market for CISL Biotechnology.
What are the main advantages for both companies?
CorePath is always looking for having academic excellence into the private enterprise. And in addition to the diagnostic side of the testing for typical cancer, we have been looking for any novel protein for detecting early cancer, any type of cancer, especially lung cancer, with this partnership. So our plan is to have an alliance together to validate this particular test that we have, which is a very simple test, and go through the validation process and other compliance requirements that we are required to go in the US, and eventually offer this testing to patients in North America, and using this model in North America can be offered to offer the test in rest of the countries utilizing their own compliance model.
What's the main issue around diagnosis at the moment? What is it that you're going to be picking up that other companies, might not have been able to do? And what does early diagnosis mean?
Hi, so this is Celina Villa. I'm a pathologist and the medical director of the Biopharma Division here at CorePath. Currently, the only recommended screening test for early lung cancer detection is the low-dose CT scan. The U.S. Preventative Services Task Force recommends yearly low-dose CT scans for people who are between the ages of 50 to 80 years old, are current smokers, or have quit in the last 15 years, and have at least 20 pack year smoking history. A pack year refers to the number of packs of cigarettes per day multiplied by the number of years smoked. For example, somebody who smoked 1 pack a day for 20 years has a 20 pack year history of smoking, as does somebody who smokes 2 packs a day for 10 years. And this recommendation was based on results of the National Lung Screening Trial that showed that screening using these low-dose CT scans reduced mortality compared to screening with chest X-rays. However, there are several disadvantages of using these low-dose CT scans. They have a high false positive rate. So in other words, the test may detect a lung nodule that turns out not to be cancer, but that still requires additional testing, such as more imaging studies or a lung biopsy to determine the nature of the nodule. In the National Lung Screening Trial, these low-dose CT scans had a false positive rate of 96%. So in this study, a lung nodule was detected in 1 of every 4 subjects. These subjects underwent follow-up tests, including additional imaging studies and biopsies, and only 4% were confirmed to have lung cancer. Another disadvantage is that these low-dose CT scans expose patients to small amounts of radiation with each test. They are low dose, so they have 5 times less radiation compared to the conventional CT scans. However, those patients needing additional CT scans will undergo more radiation exposure. There's also the added risk of potential complications for those who undergo needle biopsies or surgery. Another disadvantage is the excess financial cost that results from follow-up diagnostic procedures, including imaging and invasive procedures. As far as serum markers, they have been studied extensively in lung cancer, but no tumor biomarker is currently available to detect early cancer. So in essence, there is a real need for an accurate and cost-effective blood-based screening test that reduces unnecessary exposure to radiation and also invasive diagnostic procedures.
For somebody who may have fear that they are at risk of lung cancer and they wanted to know how your test works, how would you sort of walk them through, getting from the doctor to your test?
Screening is recommended for, like I said, people at high risk, including smokers with the history that I just mentioned. This test could have several potential uses. It could be used as the initial screening test for those patients, or it can also be used in combination with these low-dose CT scans. The advantage would be that the blood test would help rule out those patients that did end up having a lung nodule identified on the CT scan. So more studies are needed to figure those details out.
So where are you at this precise moment in the partnership, and what's your timeline, your sort of targets, and how long you're kind of going to be working together? Is it sort of ongoing well into the future, or is it sort of like a length of a project?
For any cancer test or any test which is a new test, and you are applying that test for the clinical use, there are multiple stakeholders involved before we make the test available for general population. First of all, patients themselves, a good number of patients need to be tested, normal patients as well as patients with cancer. It needs to be coordinated with clinicians and clinical-based guidelines through different organizations such as ASCO. It needs to be validated by pathologists. In our case, CorePath is taking the lead in validating the test. The validation of the test itself for clinical use requires certain compliance aspects, certain understanding by the payers to approve the test for reimbursement. It's a process that we are going to be initiating, and the timeline based on our experience and based on understanding all these steps involved can be anywhere from 1 year to 2 years before we can offer the test clinically for patients.
Are you looking to develop on from tests for lung cancer? Have you got any other types of cancer that you'd like to form a similar partnership for in the future?
As you know that any patients who get diagnosed with cancer of any type, the first question they ask is, I wish I could have been detected this earlier. Is there any simple test? So scientists and researchers have been working hard to develop a single test that can detect most cancer. Since this novel protein is specifically going to be tested for lung cancer, we have heard and we have seen that this protein can cross-react with some other cancer type as well. So our pilot is going to be focusing more on lung cancer, and we are hoping that new biomarkers are going to be emerging and can be validated in the clinical setting so we can apply a single blood test for most of the cancer for early detection. There is one point I really need to emphasize when we are dealing with cancer patients as a pathologist and or as a family caregiver. We not only deal with a number of patients. Our CorePath, just like any other diagnostic lab, is really passion-driven for caring for lives. We believe that these cancer patients are not just numbers, these are lives which are present on the other side. So the more stakeholders get involved in taking care of cancer patients, whether from the regulation point of view, from compliance, from payers, from clinicians, from pathologists, the winner is going to be these cancer patients. Because all of us as scientists, our main goal is to beat this beast that we have, and all of us, our family, friends, and loved ones get suffered. So that's really the challenge that we have as a scientist, and that's the challenge that we have to detect lung cancer or any other cancer, so patients don't get diagnosed very late into the diagnosis.
Next, it's two face masks. I admit that I have several of them with various designs on them, and I lose them quite often as well, although I do tend to find them again weeks later. Not that I wear them quite as much as I used to, seeing as COVID measures have been pretty much completely relaxed in the UK, but of course that could always change. Single-use masks are obviously a major issue when it comes to waste as well, so one educational establishment, Rensselaer Polytechnic Institute in Troy in upstate New York, has been addressing the issue by coming up with N95 surgical face masks that aren't just barriers to bacteria and viruses, but they also get rid of them. To tell us the story is Helen Zha, Assistant Professor of Chemical and Biological Engineering and a member of the Center for Biotechnology and Interdisciplinary Studies at Rensselaer Polytechnic Institute. I wonder if you could tell me something first about the Institute and the kind of research that takes place there.
Sure. I'm an assistant professor of chemical and biological engineering at Rensselaer Polytechnic Institute. We are a science, engineering, technology focused university in upstate New York with several hundred faculty members and over, I think, close to 7,000 students split among undergraduate and graduate students. We do have a very strong, research presence spanning everything from biotechnology to social science research. We are primarily government funded, so funded by various U.S. grants as well as some industry funding. So we are very, very much focused on innovation across a wide variety of fields, spanning biotechnology to artificial intelligence. The schools of engineering and science and architecture really take up numbers-wise a large portion of the university, right? There is a lot of fundamental research that occurs here, looking at really basic concepts of how things in nature work, as well as more translational technological development that occurs. And so my group personally spans the spectrum of that. We fundamentally try to understand how materials that occur in nature work, but we also use that knowledge to develop innovations that may be, for example, commercialized.
And I guess we're talking about face masks today, and everybody in the world, I think, probably has a face mask right about now. But what was it that prompted you to start to investigate this issue?
So if you remember when COVID flared up in the early 2020s, in the US, there was a huge shortage of N95 face masks for frontline healthcare workers, to the point where healthcare workers needed to jury-rig their own solutions, like putting bags over their faces and wearing multiple layers of masks. And all of these were very inadequate. And the shortage of N95 face masks was due to a lot of things. But I was having a conversation with a friend of mine who is a doctor in a medical setting, and she described to me how these masks are used, not just N95s, but even surgical masks and anything used in a healthcare setting. So the idea is as a nurse or a doctor, you're supposed to put a clean one on right before you go into a patient's room. And in the process of talking to the patient, you assume that the mask has become contaminated with the virus, which is expelled into the air by the sick patient, even in just a simple process like talking. And you're supposed to remove that mask before seeing the next person, the next patient, or maybe your next nurse, because it's assumed that that contaminant is there, and there's nothing the mask does to kill this virus. Or, if it's a bacteria, it doesn't kill the bacteria. It simply traps these microbial particles in the filter. And that's why, in order to avoid cross-contamination, you're really supposed to just they're single use. You're really supposed to discard them very, very regularly. And that goes for N95 masks, it goes for surgical masks, and even for the home user, we all know that, you really should be washing these. If you're using a cloth mask regularly, or if you're using the plastic mask, the blue ones, you should be tossing those quite regularly. There's guidelines about how often you're supposed to toss them. And that's because they don't decontaminate themselves. So they're essentially trapping live microbes that can still infect. And so we were motivated by this problem, which we saw as contributing to shortage, as well as substantial plastic waste. I mean, all of these masks are made out of non-biodegradable plastic. So we wanted to come up with a solution where essentially the mask would deactivate and kill microbes on contact. So they're designed to trap microbes as you breathe in, but we wanted them to additionally kill them so that you could wear the mask longer, potentially see multiple patients or talk to multiple people, wearing the same mask, and not have to worry about cross-contaminating people carrying that microbe, in the presence of different people. And that was the motivation for our project. And so we applied for a National Science Foundation rapid grant, which is one that is given for sort of emergency scenarios that need quick innovations. So that's the starting point of our project.
Was that right in the middle of the pandemic? Because obviously
That was in April 2020 was when we first got this grant, and it's a project that we had been working on basically since summer 2020.
you have the idea, but then you have to make it happen. How did you put a team together, and how did you work on the project?
Yeah, so my collaborator Ed Palermo and I have complementary expertise. One of my expertise areas is in the characterization and modification of surfaces. For example, you take a surface of a fiber, that's what the filters are made out of, and you modify it to have something that kills viruses. And my collaborator Ed Palermo, who is an associate professor in material science department here, is an expert in antibacterial polymers. And we had the idea about the platform and the chemistries of how this would work, but we needed some other expertise on the team. We needed people who were very familiar with assaying the viability of viruses to basically test whether or not we were actually killing viruses, different strains of viruses. We needed people who had expertise in testing the breathability and filtration efficiency of masks, which was fairly new to both Ed and I. And we needed basically help in these other areas, so expertise in these other areas. So we assembled quite a large and diverse team. We partnered with AFOA, the Advanced Functional Fabrics of America, which is an organization that it's actually a partnership that includes MIT, and they tried to advance textile innovations, essentially. So we partnered with them, and we also received a really kind gift from Hills Inc., which is a company that they are, I believe, the only company in the States that make the equipment that is used to make the filters that go into N95 face masks. So these are very specialized filters, and the material does get made overseas, but in the US, they make the equipment to make this mask. So they donated to us a large amount of just this raw filter material for us to work with, so that we can actually do all of our testing and development on the proper material that you actually find in N95 face masks. We collaborated with Greg Relich's group at MIT in the Department of Chemical Engineering. His group is an expert in testing filtration and breathability of things like face masks. And we also partnered with MIT Lincoln Labs to do some antiviral studies, as well as with the group of Karen Helt in Michigan Tech University, also to do antiviral studies to really lay down the scientific foundation and prove that our material was killing viruses, which is actually a non-trivial thing to do.
Yeah, and I guess it's not just the actual denaturing of viruses or the killing of bacteria. It has to be practical as well. It can't be something that is so big that it won't fit on your face, or that it's so big that you can't work with it or breathe through it. So it's the practical issues as well as the scientific ones.
Yeah. And, we had a couple goals in mind. One, which is that we would work with already made masks and mask materials so that you don't have to, for example, the process we developed can be applied directly to an already made surgical mask. It can be applied using very, very simple laboratory equipment and very, very widely available solvents, like acetone is a solvent that we use. It's, something you could get at home. And we wanted to use chemistries and chemicals that were also quite scalable and readily available. The reason why we wanted to do all these things and work with already made materials, like already made filters, as opposed to creating process that would have to be implemented at the point where the filter was being made, is to increase the ease of adoption, by the industry. So people who are already manufacturing face masks, people who are already manufacturing filters, can essentially add this as an additional step. They don't have to go back and retool their processes for making product as much as if we were to say develop something that you would have to implement in with their manufacturing process at the very beginning.
And I guess the other thing is sometimes science can move a little slowly, but this is something that had some kind of urgency to it. So time was of the essence here as well.
Indeed. And so that's also why we put together this very large and diverse team, because we knew that we wouldn't have the bandwidth to do everything ourselves, nor do we have the exact expertise to do it to standards which they need to be done. So the collaborative environment that we have here at RPI and also with all of our collaborators was very critical.
Could you explain a little bit about the UV technology that's involved here?
Yeah, so many people are familiar with UV as a method of actually killing bacteria and viruses, and indeed the UV can do that. But what the UV in our system does is it activates a molecule called benzophenone, and that molecule we have essentially attached to our antiviral portion of the molecule. So the UV light causes this benzophenone to react with the surface of the polypropylene that the mask is made out of. And it causes this chemical reaction, which effectively grafts or connects very strongly the antiviral polymer onto the surface of the mask. And this means that this layer, essentially this coating we've created, is very, very thin, but also it's non-leaching. So it's not like a nanoparticle that gets just physically sort of embedded and can slowly leach out over time. This is a non-leaching antimicrobial coating, which is very, very thin and covers the surface of all the fibers in these filters that you have in your face masks. And the UV is the way that the chemistry gets triggered to get that to occur. But at the same time, it's also actually disinfecting the mask.
And how was it tested? How did you test this?
We ran a number of tests on these materials. We did a number of chemical analysis, surface-based, as well as looking at properties of the mask with and without the coating. We made sure to test the filtration efficiency of the mask before and after applying our process, as well as the breathability of the mask. So there's actually equipment and criteria that needs to meet. We also did a variety of antimicrobial tests. So we tested the ability of the mask to kill gram-negative and gram-positive bacteria, which it was able to do so with greater than 99.9% efficiency. And we also tested its ability to deactivate, kill viruses. Actually, we used 3 different strains of envelope viruses. So coronaviruses are a specific kind of virus called an envelope virus. And so we tested with 3 different strains of virus. We didn't test with human coronavirus. We don't have the facilities to work with that virus, but we did test with several viruses that are very similar in structure, and they themselves actually are in the coronavirus family. So we wanted really to make sure that this material was actually killing these viruses, and we achieved up to 99.99% killing efficiency of these kinds of viruses with our material. And so we were very comprehensive in making sure the boxes were checked, that it does kill bacteria and viruses, that it is still breathable, and that it still has the filtration properties that you'd want.
And I guess one of the big pluses about this system is that there's far less waste involved.
Yeah, so that's one of the, I think, the benefits of having a self-decontaminating mask, whether that comes from our innovation or, there are other innovations out there that are trying to do something similar, is that you can reuse the same mask. Whether that means you can just wear it longer before you have to discard it, or you can actually potentially even decontaminate it, or it's just self-decontaminating. You can wear it for a lot longer, which means, instead of a doctor going through, 20 masks a day, maybe they might only need one or a few. And so we're really reducing the amount of plastic waste, hopefully, with technologies like this. And hopefully also reducing, shortages in times like a sudden increase in need.
And I guess though, once you have a product like this, it then goes on to the next stage, which is kind of the commercialization stage. How are these going to be manufactured, and is it going to be something that's economical to do?
Right. So we specifically targeted, I think, very chemistries with potential to be scaled easily, and processes and that can be scaled, such as the UV irradiation, as well as solvents that are pretty inexpensive, right? Like acetone. In a very simple method. So the goal of which was to make it readily commercializable. Whether it will be cost effective, I think that depends on a number of things, which at the academic level, we're not pursuing understanding of that. Right now, these masks are dirt cheap, right? They're designed to be essentially the equivalent of a plastic spoon that you get for less, pennies and you toss. And certainly adding this kind of feature on top will increase the cost, but will that be offset by less waste and, essentially the need to use less of them? That's the kind of techno-economic analysis that anybody who wants to commercialize this would need to do. We published our work open access, meaning that we made it available to anyone and everyone to download this without having to pay anything for the article. And Ed and I are always happy to, discuss further with anyone interested in trying to commercialize and optimize production for this technology. At the academic level, it's not something we are currently working on. We create innovations and we kind of let that information out into the world. And we have our expertise, which we can always are willing to help. But I definitely think there's potential for this to be commercialized. And we specifically, wanted to design the processes in a way that would really facilitate that.
In the interim, we've moved to a situation now where not so many people are wearing masks. Would this still be applicable for, in hospitals where people would wear masks, and also preparedness for, I hate to say it, but there will be other pandemics in the future?
Yeah, absolutely. This is a broad spectrum acting material, which means it kills many, many strains of bacteria and many, many strains of viruses. So even if COVID wasn't an issue, this still would be very beneficial in a hospital setting where people will always need to be wearing some sort of face PPE, personal protective equipment, doctors and nurses and patients, whether that's protecting against even the simple flu. So I think this will definitely be useful in that scenario, even not in the scene, of a pandemic, right? It will still have the ability to increase protection and decrease waste and increase the usage lifespan of masks. For the personal user, there is a lot to be said, I think, moving forward about being more aware of, there are many cultures in Asia, for example, societies that wear masks sort of on a seasonal basis. And it has nothing to do with the coronavirus or COVID, but it is a good way to protect yourself during flu season, for example. So it's still useful, I think, even for the personal user as a part of their lives. And one other thing that we're interested in is expanding this technology to filters that are designed to purify, our living spaces. So HEPA filters are a good example. They're actually very similar in technology to the filters that go into your face. They just have to work at different specifications, essentially different airflows, but they're still fundamentally made out of the same material. We can imagine applying antimicrobial coating that deactivates any bacteria and viruses that caught in your HEPA filter from your room. This is particularly important for enclosed spaces, I think. For example, submarines, ships, elevators, airplanes, etc. Right? Having this active decontamination, I think, will really enhance the air quality, essentially, of these spaces. So this is definitely, I think, an area that will grow in need. and their innovations are going to be relevant moving forward, regardless of where we stand in terms of pandemics and COVID.
You mentioned the fact that this is open access and open to anybody and collaborations. What are the next steps for this? Is it really just sort of waiting to see who approaches you about it?
Right. We are definitely open to collaborations, utilizing the expertise that we've gained as well as the team we've built on this project to move forward with either continuations of this project or diversification to other filtration systems, for example, HEPA filters. Or even we can think about filters that are used for more niche applications like purifying pharmaceuticals in their manufacturing process. They could actually really use something that kills viruses. So we're actively seeking funding opportunities for some of these ideas. We're actively seeking collaborations, and we're, of course, very welcome to anybody contacting us if they are interested in working with us, if they are interested in commercializing and would like us to be involved in some way. Pretty much open to anybody and their ideas and working with them, yeah.
And there you have it, another Beyond Biotech podcast, another week over. I'm still allegedly on vacation next week, but it's just day trips with the family, and of course it's all weather dependent, which is a big challenge in a country where you can experience 4 seasons in one day. Because I'm not working as much, all of the interviews for the next podcast are already done, but sadly they are also not edited yet. So next week we will have conversations with LifeLength, ISA, and a Neurotech. As always, if you do have a press release for us, or someone you think we should feature on the podcast, we are very happy to hear from you, and you can drop us a line through the Labiotech.eu website. All right, I hope you have a great week ahead, and we thank you for listening, and hope you'll join us again next time, wherever in the world you are, for the next Beyond Biotech.